All
so
right,
cute.
so our group is going to be talking about fetal development from our textbook, chapter 12. So our objectives for this PowerPoint are to discuss the significant changes in growth and development of the embryo and fetus. We will contrast the different types of multifetal pregnancies and analyze the potential effects of teratogens during vulnerable periods of embryonic and fetal development. All right, so we're going to be starting out with fetal maturation. So the actual fetal stage is going to last from 9 weeks up until that pregnancy ends. There's a great table in our book. It's table 12.1. It goes through every stage for each week and the changes that happen. And really, most of the changes are going to happen in that embryonic stage before. So during the fetal stage, the changes really aren't that dramatic. It's really just a period where everything becomes refined of structure and function. So when we're talking about viability of a pregnancy, this is defined as the capability of the fetus to survive outside of the uterus. And this definition is going to vary from state to state, but typically it is defined by the fetal weight and the pregnancy duration. So a standard definition of this is going to be 350, 400, or 500 grams for the weight and 20 weeks gestation. But again, that does vary. With advances in technology and science, it's really more that infants who are 22 to 25 weeks of gestation are really within that threshold of viability. And on the other side of this sheet here are chemical teratogens, I had to Google it, that happen or that can really affect the embryonic and fetal development. So this list, it does cover a lot that's in our book, but our book covers just a little bit more. And looking through these, again, they really don't affect as many or as much during that fetal stage, but the embryonic stage is where most of these are affected. Really the only chemicals that are going to affect the fetal development are teratogens that cause malformation or affect the functioning of the central nervous system. Okay, fetal circulatory system, this is pretty cool. So the cardiovascular system is actually the first organ system to function in the developing human. So the fetal lungs actually aren't functional during that period. They function at birth. So how does the fetus get its oxygen and nutrients and everything it needs to survive? Well, it actually comes from the placenta. So the placenta is going to provide oxygen and nutrients to the fetus, and it's also the source of gas exchange. Now, the umbilical cord has one umbilical vein, and that is actually going to carry oxygenated blood from the mom to the fetus, and then 2 umbilical arteries that's going to carry away the deoxygenated blood with the waste from the fetus back to the placenta. So the oxygenated blood is going to flow from that umbilical vein. It actually goes to the fetus's liver. And the liver's not fully functional, but it does have some blood flow. So what happens within this fetal system is there are shunts that actually bypass a lot of the structures that are not really needed during that period. So the liver and the lungs. So when it goes to the liver, it will use some of that blood, but then it will be shunted via the ductus venosus to the inferior vena cava. And from there, the blood flows into the right atrium, and then it's shunted to the left atrium via the foramen ovale, and that connects, that connects the right atrium and the left atrium. It goes down to the left ventricle, then through the aorta to replenish the fetus body. Now, simultaneously, deoxygenated blood is flowing in from the superior vena cava, And most of this blood flows into that right ventricle. It is pumped through the pulmonary artery, and some of the blood will be sent to the lungs, but that's really just for tissue growth and health. Most of that blood is going to be shunted via the ductus arteriosus from the pulmonary artery into the aorta. And this blood, some of that blood will go towards the fetus limbs, but the descending aorta also branches off to the internal iliac artery, which connects to the umbilical arteries, and that's going to carry back that deoxygenated blood to the placenta, where it will go into mom's circulation. From there, she will rid the blood of waste and then send back oxygenated blood to the placenta.
Wow.
Pretty cool. I know, isn't that awesome? Okay, and then speaking of respiratory system, it is also developing during this period. So weeks 4 to 7, it's the development of the larynx, the trachea, bronchi, and lung buds. So weeks 16 to 24, the bronchi and terminal bronchioles enlarge. And then 24 weeks to term gestation, just more alveoli are forming. Now, it is necessary that pulmonary surfactants are also being built during this time. This is going to help that baby breathe and their alveoli to open up after birth. So there are type 1 and type 2. And after 32 weeks, there should be sufficient surfactant present. And it is used to, this LNS ratio is used to determine the degree of fetal lung maturity or the ability of lungs to function after birth. So the lecithin is the most critical alveolar surfactant required for that lung expansion. And then the sphingomyelin is a phospholipid that is just always constant. So what we would be looking for would be a 2-to-1 ratio, the L to the S. That's when the infant's lungs would be considered mature. And that usually does occur in the middle of the 3rd trimester. There are some conditions that could inhibit lung maturity, gestational diabetes and chronic glomerulonephritis. And then there are also conditions that will accelerate lung maturity, and that's maternal hypertension, placental dysfunction, infection, or corticosteroid use. Another interesting fact, shortly before birth and during labor, the lung fluid decreases, and then after birth, the remaining fluid is reabsorbed into the infant's bloodstream. But neonates born by C-section birth, scheduled C-section, before 39 weeks gestation without labor have an increased risk of transient tachypnea and are more likely to have crackles in their lungs for the first 24 to 48 hours. I like that fact. Okay.
It is.
Okay.
All right. Okay. So I have the gastrointestinal system. So the foregut, well, mind you, during the 4th week, the shape of the embryo changes from being almost straight to like a C shape as both ends fold towards more of the ventral surface. So this is where the 3 factors come in. A portion of the yolk sac, which is a weird name in my opinion, is incorporated into the body from head to tail as the primitive gut. So the foregut produces the pharynx, part of the lower respiratory tract, the esophagus, the stomach, the first half of the duodenum, the liver, the pancreas, and the gallbladder. So these structures evolve during the 5th and 6th weeks. Malformations such as esophageal atresia, hypertrophic pyloric stenosis, man, that's a long word, duodenal stenosis or atresia, and biliary atresia can occur. Moving on, you got the midgut. The midgut becomes the distal half of the duodenum, the jejunum, and the ileum, the cecum and the appendix, and the proximal half of the colon. The midgut loops projects into the umbilical cord between weeks 5 and 10. Malformations can result if the midgut faults to return to the abdominal cavity, so it like folds and all, causing the intestines to protrude from the umbilicus. So think of like mecodiverticulum is the most common malformation for the midgut. It occurs when a remnant of the yolk stalks that fail to degenerate attaches to the ileum, leaving the blind sac. And then lastly, the hindgut, so think of like the body from top to bottom, develops into the distal half of the colon, the rectum and part of the anal canal. the urinary bladder, and the urethra. And anorectal malformations are the most common abnormalities of the digestive system. So again, anorectal, easy stuff. And the hematopoietic system is the formation of blood, which occurs again with the whole yolk sac situation, beginning of the 3rd week. As you can see from the diagram, stem cells seed the fetal liver during the 5th week, and hematopoiesis begins during the 6th week. This accounts for the relatively large size of the liver between the 7th and 9th weeks. And stem cell seeds the fetal bone marrow, spleen, thymus, and lymph nodes between weeks and 8. So you have the two differences between spermatogenesis and oogenesis. Great.
Hi guys. So the hepatic system. So by the 4th week of gestation, the liver and the biliary tract develop from the foregut. And then during the 12th week of gestation, bile begins to form. And then at the 9th or 10th week of gestation, glycogen is stored in the liver of the fetus. And at term, glycogen stores are actually 2 times of those as an adult. And that's because glycogen is a major source of energy for the fetus. So it makes sense. Iron is also stored in the fetal liver. And they can store enough iron to last 5 months after birth. And that's only if maternal intake is sufficient, so it's really important to educate the mother on proper intake of iron so that your fetus can be healthy. During fetal life, the liver does not have to conjugate bilirubin for excretion because the unconjugated bilirubin is cleared by the placenta. So the placenta kind of does the work for the fetus. Glycuronol transferase enzyme needed for conjugation is present in the fetal liver in amounts less than those required after birth. And so we kind of talk a little bit about hyperbilirubinemia, and this predisposes the neonate to that because, like I said, the amounts are less than what they actually need. So these coagulation factors 2, 7, 9, and 10 cannot be synthesized in the fetal liver because of the lack of vitamin K synthesis. So we kind of talk about prophylactic administration of vitamin K to the newborn. We talked about like giving that IM shot to them. And so this is the reason for that, because those coagulation factors can't be synthesized. Next slide, renal system. So the kidneys form during the 5th week of gestation and begin to function at approximately the 9th week. So think 5th week, kidneys form, 9th week, kidneys function. Urine is excreted into the amniotic fluid and forms a major part of the volume. So we learned about oligohydramnios, and that's lack of amniotic fluid. And so that's indicative of renal dysfunction, because if they don't have enough amniotic fluid, it's probably because urine is not being excreted, since it makes up a major part of it. The placenta acts as the organ of excretion and maintains fetal water and electrolyte balance. We know this, the placenta helps the baby a lot. So the fetus doesn't need functioning kidneys while in utero. But while at birth, the kidneys are required immediately for excretory and acid-based regulatory function. So they don't function while in utero, but they're needed at birth. A fetal renal malformation can be diagnosed in utero, and corrective or palliative fetal surgery may treat the malformation. But there can also be treatment plans made for immediately following birth. So like I said, at term, the fetus has fully developed kidneys. They're not needed during utero, but they're needed at term. If the GFR is low, the kidneys lack the ability to concentrate urine, and so this makes the newborn more susceptible to both overhydration or dehydration, just because like the urine it can either be too concentrated or not concentrated enough. Most newborns void within 24 hours of birth. With the loss of the swallowed amniotic fluid and the metabolism of nutrients provided by the placenta, the amount voided for the first few days is scanty until fluid intake increases. So the 3rd week after fertilization, this is for the neurologic system. The 3rd week after fertilization, the nervous system originates from the ectoderm. 4th week after fertilization, the open neural tube forms, and the embryo folds in on itself lengthwise, so like that. And it forms a head fold in the neural tube. The cranial end of the neural tube closes, and the caudal end also closes. So the 5th week after fertilization, different growth rates cause more flexures in the neural tube, so like more folds, and this delineates 3 brain areas, so the forebrain, the midbrain, and the hindbrain. So let's talk about the forebrain. Cranial nerves 1 and 2, which is olfactory and eyes. Olfactory is 1, eyes is 2. The cerebral hemispheres and the thalamus, that's what makes up the forebrain. The development of all areas of the cerebral cortex continue throughout fetal life and into childhood. The midbrain is cranial nerves 3 and 4, and that's oculomotor and trochlear. And then the hindbrain is made up of the medulla, the pons, the cerebellum, and the remainder of the cranial nerves. So all the rest of the ones I didn't mention. Also, fun fact, brainwaves can be recorded by week 8. The spinal cord develops from the long end of the neural tube, and the neural crest develops into the peripheral nervous system. By the 8th week, nerve fibers transverse throughout the whole body. At term, the fetal brain is 1/4 of the size of an adult brain, so it's really tiny. Neurologic insult can result in cerebral palsy, neuromuscular impairment, intellectual disability, and learning disabilities. So the sensory awareness of the fetus, firm touch to the mother's abdomen, should elicit movements from the fetus. By 24 weeks, the fetus responds to sounds. We learned a little bit about acoustic stimulation and how it should evoke a fetal heart rate response. 16 weeks, taste receptors are present so they can actually taste the maternal diet, which is really interesting. By the 5th month, a sweetener added to the fluid causes the fetus to swallow faster, while a cold solution placed in the amniotic fluid can cause hiccups, so your little fetus can be hiccuping. Eyes are fused until 24 to 26 weeks, and by the 7th month, rods and cones are formed in the retina.
All right, that's me again. Endocrine system, and there are 3 functions within the system. So we have the thyroid gland, which develops along with structures in the head and the neck during the 3rd and 4th weeks. This includes the secretion of thyroxine begins during the weight week. Maternal thyroxine does not readily cross the placenta. Therefore, the fetus who does not produce thyroid hormones will be born with congenital hypothyroidism. And if it's left untreated, it can result in a severe intellectual disability throughout its span. Then the adrenal cortex is formed during the 6th week and produces hormones by the 8th or 9th week. As the term approaches, the fetus produces more cortisol, and this is believed to aid the initiation of labor by decreasing the maternal progesterone and stimulating the production of prostaglandins. Then the pancreas form from the foregut during the 5th through 8 weeks. The islets of the Langerhans develop during the 12th week, and insulin is produced by the week 20. In fetuses, as we all know, if mothers with uncontrolled diabetes, maternal hyperglycemia produces fetal hyperglycemia, which stimulates the hyperinsulinemia and islet cell hyperplasia, which again results in macrosomic large fetus. And the hyperinsulinemia also blocks lung maturation, which can lead to respiratory distress.
Alrighty. So, reproductive system. Sex differentiation begins in the 7th week. By week 9, genitalia, male and female, appears and is fully differentiated by week 12. So females, that's up there, right? Now it's
No.
Oh no, I went back.
You're
So
good.
sorry.
It's the other one.
Other computer.
Other computer.
Oh, so I was sorry. Continuing. So, the female has 2 X chromosomes, and that, of course, makes the ovaries and the female external genitalia. Oogenesis, this is really cool. The baby, when it's born, has lifetime supply of the eggs that it's going to have the rest of its life, and that has been established at the 16th week. Most female hormone production is delayed until puberty, which is different than males, which low levels of testosterone continue until they hit the pubertal surge. And female babies can have something called pseudomenstruation at birth, which is like a baby period. It's kind of interesting, which is when a lot of hormones are lost at birth, maternal hormones. So it's like the baby's response to that. And with the males, the Y chromosome is present, which forms the testes. And by the end of the embryonic period, testosterone, like I said, is being slowly secreted until the pubertal surge. And by 28 weeks, the testes began descending into the scrotum. And something interesting, high level of maternal estrogen stimulates mammary engorgement and secretion in both sexes, so males and females. So musculoskeletal system, bones and muscles begin forming in the 4th week of embryonic development, which is so early to me. That's pretty crazy. So this is pretty much the progression of development. The mesoderm next to the neural tube forms the vertebral column and ribs. Parts of the vertebral column grow toward each other to enclose the developing spinal cord, and ossification, bone formation begins. Spina bifida, neurologic deficits, and skeletal deformity can occur if there's a deficit in bony fusion during this time. By the 6th week, bones of the shoulders, arms, hips, and legs appear as a continuous skeleton with no joints. So no joints have come together at the 6th week. Differentiation occurs, which then produces separate bones and joints, and ossification continues. By the 7th week, muscles begin to contract spontaneously, and by week 11 and 12, the fetus makes respiratory movements, moves their extremities, and changes position. They can suck their thumbs, swim around, make loops in the umbilical cord, which is really crazy. And those arm and leg movements are visible in the ultrasound, but the mother can't detect them until like 16 or 20 weeks of gestation. So this is a picture of a fetus at 11 weeks in utero. And the integumentary system, I don't know if that's how you say it. So the epidermis, by 4 weeks, there's just a single layer of cells. By 7 weeks, there's 2 layers. And by 7 weeks, this starts to form the vernix caseosa, I think is how you say it. So this is like the cheesy stuff that is on the baby when they're born. It's like helps protect their skin. That begins to form in the 7th week. The basal layer of the epidermis is called the germinal layer, and this replaces lost cells. Up until the 17th week, the skin is wrinkled and like weird looking, but then subcutaneous tissue begins to form, and that makes it like not as wrinkly and red and stuff. That's what makes the baby like more plump. At term, that's when the skin starts to thicken. At 32 weeks, it's less wrinkled and red like it would be in utero. It would be like really wrinkly, and lots of blood vessels are visible. And by 16 weeks, the epidermal ridges are present. The baby's hair begins to form by bulbs in the epidermis that project into the dermis. Lanugo is like the really, really thin layer of hair all over the baby. So at 12 weeks, it's on the eyebrows and the upper lip. 20 weeks, it covers the entire body, including the eyelashes, eyebrows, and the scalp. And then by 28 weeks, the hair is longer than like standard lanugo, so it's like normal hair. Fingernails and toenails develop from thickened epidermis at the tips of the digits beginning during the 10th week, and then the fingernails usually reach the fingertip by the 32nd week, and toenails reach toe tips by the 36th week. Super specific, but pretty cool.
Right, hello. So, wrong way. So, the immune system. During the 3rd trimester, albumin and globulin are present in the fetus. Passive immunity occurs with IgG, which is an antibody that crosses the placenta. The next time the passive immunity occurs is after birth, when the infant's being breastfed through colostrum, which is also known as liquid gold in baby world. And it comes before like the production of regular breast milk. At the end of the 1st trimester, IgM, another antibody, is produced in response to the blood group antigens, gram-negative enteric organisms and viruses, and preterm infants are more at risk for infection than normal term births. Oh, wrong way again. Multifetal pregnancy. I just put some pictures up here so you guys could see like what a fused placenta looks like, and just some easy photos to help memorize some things. But there are two types of twins, dizygotic and monozygotic, I think is how it's said. Dizygotic is fraternal twins, so there's 2 amnions, 2 creons, which you can see in this photo what that is, and 2 placentas. They can be separate or fused. They can be the same sex or different sexes, not genetically the same. And then there's some risk factors for dizygotic twinning, and it occurs in 1% to 1.5% of natural conceptions. And then you have the monozygotic twins. Those are the identical twins. Although they are identical, it doesn't mean that their genetics are exactly the same. Cell division is between 4 to 8 days. After fertilization, there are 2 embryos, 2 amnions, 1 creon, and 1 placenta. It occurs in 0.4% of pregnancies, and use of ART, known as assisted reproductive technology, increases the incidence of monozygotic twinning. And conjoined twins or Siamese twins are a type of this kind of twin which cleavage is incomplete and it occurs more than 13 days after conception. Triplet or high order births, basically triplets is like 1 zygote divides into 2 and one of the 2 divides again and makes an identical triplet. There's other ways that can occur, like 2 zygotes produce a triplet. 1 zygote divides and makes a set of monozygotic twins, while the second zygote develops a single fraternal sibling, or from 3 zygotes. It's kind of confusing, but just crazy how that happens. And then quadruplets, quintuplets, sextuplets, they divide in similar ways. And then there's just another picture for you guys to see the monozygotic twins. And then factors influencing fetal growth and development, complex interactions between genetic and environmental factors. Let me get this slide over here. Fetal parental genetics, congenital anomalies and disorders are a result of chromosomal abnormalities known as teratogens. I think that's how we said it. And I put the table 12.2 on this slide so you guys could see some teratogens cause diseases that cause congenital effects. And they're produced by environmental factors. So it really depends on the time in pregnancy that the exposure occurs. The most critical period for embryonic development is the first 3 months of pregnancy. The greatest effect that teratogens have on the fetus is 15 to 60 days during the embryonic period, and during the 2 weeks of development, the first 2 weeks of development, teratogens either have no effect on the fetus or the effects are so severe that mothers usually have a miscarriage of the fetus. And then those are our resources. And also, for mothers who want to track fetal development and maturation, they can download apps on their phone that can track that, such as OVA Pregnancy Tracker, Huckleberry Baby and Child, and Hi Mommy Pregnancy and Baby app. Thank you.